Right, no introduction, let's get into coronary heart disease. So, what's the problem? Blood clots formed when you get phasic deposits in your arteries. This means oxygen can't get to the cells, they die off. Unfortunately, this leaves scar tissue. Too much scar tissue, your heart fails. This means that coronary heart disease is the single biggest killer of all diseases in the Western world, responsible for 3 out of every 10 deaths. So the medical profession has approaches to try and mitigate this, but none of these actually try and deal with the actual damage done. And so this is where tissue engineering comes into play. So what we want to do is take cells from the patient, in particular stem cells and similar cells like that, modify them outside the body, increase their number, and put them back in to restore tissue function. And there's a number of different approaches that we can take to try and do this. So you may have heard of the bone acute myocardial infarction large-scale clinical trial being carried out by St Bart's and Queen Mary's. And this looks at the injection of cells directly into the heart after a patient has a heart attack. However, we find that actually the primary outcome of this is actually health and safety. So we don't know what effect this will have on the body, and so actual improvement in the heart is actually a secondary outcome for this. And the reason for this is that in rat models, you find that injecting the cells into the heart, 2 weeks later, 5% of those cells have survived, 92% are dead, and 3% are found in other key organs. And we don't know whether that's going to have more effects for creating other diseases or have other health issues. So another option is to use cell sheets. We grow the cells in the lab, and then we can layer these cells together to create the tissue that we want. However, there's a limitation in the maximum thickness that we can actually achieve. We find that this maximum achievement is about 70 to 80 micrometers, after which cells in the middle start to die off. And this is a problem because your heart wall can reach up to 1 centimeter in thickness. And the problem here is that essential nutrients and oxygen can't actually reach these cells in the middle to actually let them grow. And that's because we're not getting capillary growth. And so this is something that needs to be addressed in order for this kind of technology to work. And there is also another third option that we can use, which is decellularized heart tissue, which will appear very shortly, I'm sure. Here we are. So on the left here, what we've done, we've taken an organ donor's heart, we've removed all the cells from this donor, and so you get the extracellular matrix remaining. And this is essentially the natural scaffold for cells. We can then put new cells in there from the patient and put that back in. But we find these nice white columns here. We get a decrease in the tensile strength of the material. And we also lose key molecules from this. And this can affect your cell growth and your tissue function afterwards. So this also needs to be addressed. So the fourth option, which I'm interested in, is prefabricated scaffolds, where we put the cells onto the scaffold. And this means the cells should be retained, and we can also promote specific growth for particular cell types. But there's a number of properties which we need to control for this. So, for example, tensile properties. It's been shown that your stem cells will differentiate into different cells depending on how elastic that material is. So for very stiff material, you get bone cells, and for very elastic material, you get nerve cells being produced. Also need to look at chemistry. So your functional group will influence how well the cells attach to your scaffold, and you also want to look at incorporating growth factors to ensure that the stem cells differentiate to the cells you want at particular times. Another option is biocompatibility. So to summarize, Williams, if the material is going to cause more harm than good, don't put it in your body. Quite simple. But you've also got to look at, particularly in polymers, how this biodegrades. So on the left, we have some polymers which are biocompatible. And on the right, we have some materials from these polymers that actually can cause problems in the body. But this isn't an issue if your body can metabolize them faster than they're produced. And this is where rate of degradation comes into account. So we want our material to degrade at the same rate as the new tissue and extracellular matrix is produced. And this means that we can go from our graft to healthy, functioning material, tissue without any problems. And finally, we want to look at processability. So the process that you choose will influence the pore scaffold structure, and this can influence how the cells attach and whether the cells will align in the fashion that we want, which is particularly important in the cardiac environment because we want our muscle cells to be aligned nicely. So my work, I'm a lowly PhD student, and I'm looking at bio-based materials, in particular chitosan, and it's got a number of properties which are great, but it's got poor tensile properties. So we want to look at incorporating a second polymer into this, in my case, cellulose. Cellulose is great for tensile properties, but has two problems. First of all, it's difficult to process due to the strong bonding, and also can't be degraded by human enzymes due to a lack of functional groups. And so I'm looking at trying to address these issues, hopefully create some scaffolds, and grow some heart cells. Thank you.